So this is going to be kind of the mundane side of things. This is not future or models I've developed or anything like that. This is the consumer stuff. My somewhat contrarian take is that this very mundane stuff actually could be pretty useful. Not AGI, but helping you stress test ideas and helping shorten that wacky idea to let's run an experiment pipeline and expand the number of people who can contribute in various dimensions. So you still need to test the hypothesis, but you can change the process and broaden the process of getting there. Q, please. Oh, sorry. We just have a little lag. So the skeptical perspective is, who cares about you spend some time brainstorming with an LLM? Everything is about execution, 99% perspiration. But part of that is just because idea generation is invisible. You don't see what happens when somebody just has a blank piece of paper or their own head. And it's brief. And it determines what you do next, where you spend all your time and money. And there's this theoretical framework for the series of experiments you choose to do that determines how successful you'll be in the clinic, whether it has good predictive validity, whether you're using the right model. This is extremely impactful. But these graphs are just to the effect of, if you picked under modeling assumptions, this is from Jack Scannell, if you picked better experiments to run, you would have better ROI as a drug discovery, drug development pathway. The other thing we know very well is that methods drive biology. You invent a new experimental method, you invent a new way of collecting data, and then there's a huge amount of knowledge that follows that. And we've seen just in the past decade or two, there's a bunch of new kinds of things, optogenetics, single-cell sequencing, CRISPR, expansion microscopy, these kinds of things. And you think about what goes into inventing them. Where do they come from? Who invents new methods? Well, heavily, it's people who come from a physical science or computer science background, who have gone into biology. And in some cases, it's serial inventors. It's people who just are very good at this, and they crank out one thing after another. So that's not a lot of people. If methods drive biology, how do we make more people into methods developers? So one thing that LLMs can do is they can fill in gaps in your knowledge. So this is not about the AI knowing things that humans don't know. This is about the AI knowing things that humans who are not you don't know. The LLM is a better search engine, a natural language search engine. If I have, and this is a real life example of something I did with plain old ChatGPT a couple versions ago. I'm trying to figure out experimental design for transcranial focused ultrasound for a company that does that. Acoustics. I don't know acoustics. So I have silly questions like, would passing through the skull change the acoustic frequency? No, it doesn't. Or can you translate that experiment into literature to humans with the same parameters? No, that voltage is probably very bad for you. The analogous voltage to get the same pressure inside that location in the brain. Also things like physical limits on the technology. This is probably too small to see, but there are things like, okay, in the limit, how small and portable would these devices potentially get if you could print them out of chips? What is the limiting factor? And you ask follow-up questions, and you can get to things like, oh, the limiting factor is probably generating enough voltage in a small enough space with a smaller battery. That would be a challenge. And then things like very open-ended questions. So a couple years ago, a boss asked, so could I detect COVID-19 on a surface remotely, no labels, nothing destructive? And I thought about it, I read a little bit, my answer was no. The modalities I knew about said this, we're not going to be able to detect specific viral molecules distinct from other kinds of organic things. Infrared's not going to do that. mass spec methods would be destructive. But apparently, O3, if you ask it, will give you suggestions and will give you citations for fairly specific protein and nucleic acid peaks that can be detected with some of the newer surface-enhanced Raman spectroscopy or CARS variants on Raman spectroscopy. Does this work? I don't know yet. Would I have to consult somebody before it would be worth it to even think about investing further into the process of developing such a device? Yeah. But this really shortens the wacky idea somebody, back of the envelope one day to what would you need to find out to see if it's real. This shortens the, maybe I should make a prototype. Maybe I should start a company around this question and answer phase, and makes it accessible to more people who might not be spectroscopy experts. And this is the same problem continued of like, okay, so what molecular features can you detect with things like CARS? And it gets you some citations, and I've spot checked this stuff. It's the most literal kind of hallucination of this. The source doesn't say what the LLM says it says. That has been fairly well ironed out. Now, the subtler things like, is this a trustworthy source? You don't necessarily get that. Or is this every source? Or are there more recent things that found otherwise? That you don't necessarily get. But literally, is it in the text? If you control F, it is. So another angle is target discovery. Oh dear. My examples are not going to get finished. But targets that actually get drug developed are very restricted. They're usually the tried and true. So things like combine a big data set like Selleck's gene, look at differentially expressed genes in a disease, and then much more rapidly, this is something Future House has developed, this is the alpha version, tell me about, hit the literature, tell me about the relevance of this gene to the disease I'm looking at, and maybe get a reduced, streamlined list of genes that have some kind of known relevance to disease. And this gives you, this helps with the pen and paper speculation, what experiments should we run stage. So more people and faster connections with the existing literature, more people, you can sanity check your wacky ideas faster, which means that you can broaden your range of what you could consider. Weaknesses is it's never very exhaustive. It will include the bad research. It won't necessarily come up with the serendipity that a human expert will, and it will include implausible possibilities. It will include if you ask it to generate causes, what could cause this? It'll include kind of silly things. So really think of it more as a search engine than an oracle. It doesn't know, but it actually is what Google only hoped to be.